All right, so there's other places that other proteins could end up going. Right? So what we'll do is show you how they end up going there. So again, say that this is now a different messenger RNA, and when it's translated, it does have a signal sequence on there. And again, a signal sequence is just a particular sequence of amino acids that's found on the protein. And what that signal sequence does is that it kind of lets the cell know where to deliver this particular protein. One of the things that some particular signal sequences can do is this. So in this particular case, this protein has a signal sequence, which means that when it ends up folding, it could end up in one of two places. So there are some signal sequences that cause the protein to be delivered to the nucleus of the cell, and there are several reasons why the nucleus might require protein. So it might be a pore that's found within the envelope of the nucleus. It might be some sort of structural protein that's going to help maintain the integrity of the nucleus. So there's all kinds of reasons why a cell would be interested in having proteins in the nucleus. And again, the signal sequence tells the cell to deliver that protein to the nucleus. The signal sequence could also inform the cell to deliver it to the mitochondrion. Right? So there's all sorts of reasons why mitochondria might need proteins. Some of the proteins might act as enzymes that are required for all of the metabolic reactions that are happening inside the mitochondrion. Some of the proteins might be pores that are required to be in the membrane of the mitochondrion. Right? So again, proteins, they could end up in many different places. They have many different functions. The way that they end up going to the nucleus or the mitochondrion is that they have a specific signal sequence on there that tells the cell that that's where they belong. Right? So the bottom line is that some signal sequences, they direct the new proteins to end up going to either the nucleus, the mitochondrion, or even the chloroplast. There are different types of signal sequences, and what the different types of signal sequences do is that they tell the cell to deliver the protein to different spots. So we just saw one example of what a signal sequence can do. Here's another example. So say that this is now a different messenger RNA. Some messenger RNAs, they encode for, so say that this is being translated, and here's a ribosome. Some messenger RNAs encode for a string of amino acids, a signal sequence, that ends up having something bound to it. Right? So some signal sequences are bound to by a particle called a signal recognition particle. And basically what this particle does after it binds to a signal sequence that looks like this is that it kind of pauses the process of translation so that the protein is not completely made just yet. And what occurs during this pause, so that's the signal recognition particle is just saying, hold up, stop making the protein for a second. And what it's doing during this pause is that it allows the ribosome, with its messenger RNA in blue and its partially made protein in brown, it allows it to go to the endoplasmic reticulum. And so basically this is how ribosomes end up at the rough endoplasmic reticulum, is because of this process. This is how the ER becomes studded with ribosomes. Once it gets to the ER, what occurs is that this signal recognition particle ends up coming off at this point. The signal sequence ends up coming off at this point. And what this does is that it allows the protein now to, it allows its synthesis to complete. Right? So it was on pause for a while. Now that it has gotten to the endoplasmic reticulum, the rest of it can be produced. After the rest of the protein is produced, it ends up just getting dropped off inside of the ER. All right. So just to sum this part up in words, there are other signal sequences that cause protein synthesis to pause for a while. During that pause, again, the ribosome ends up going over to the ER, and once it's at the ER, the signal sequence comes off, the signal recognition particle comes off, and that allows the rest of the protein to be made. So once this protein has been delivered to the ER, and once its synthesis has become complete inside the ER, there are several things that could happen to proteins that are produced inside of the endoplasmic reticulum. And so we'll show you some of those things now. Right, so what all can happen to proteins that are made inside of the ER? One of the things that can happen is this. It could have yet another signal sequence attached to it. And when that occurs, after it folds, it could end up staying inside the ER. Right? So there are some proteins that have a function. They do something inside of the endoplasmic reticulum. And the way that the cell knows to keep it inside of the endoplasmic reticulum is that it ends up placing a specific signal sequence on there. So what all can happen to proteins that are made in the ER? One thing that can happen is that another signal sequence is added to it, one that'll cause the protein to stay inside of the ER. And so the job of this protein, it does something inside of the endoplasmic reticulum. Another thing that could happen to proteins that are made inside of the ER is that they could have carbohydrate groups added to them. And what occurs after this protein ends up folding is that it could be delivered to places. So an example of a place where this type of protein could be delivered is the lysosome. So lysosomes, if you remember, it's an organelle that contains a bunch of digestive enzymes. Enzymes are proteins. So if it's one of these digestive enzymes that are found in lysosomes, this is how that digestive enzyme ends up getting there. Another example of a place where this type of protein could end up, one that has carbohydrates stuck to it like this, is embedded within the membrane of the cell. So early in the quarter we mentioned that within the membrane of cells you'd expect to find proteins. A lot of them act as pores that allow different molecules in and out of that cell. So this is how protein pores end up getting to the membrane. Right? They have a signal sequence that brings them to the ER, and then carbohydrates are added to them that allows them to get now transported and then embedded within the membrane of the cell. So another thing that could happen to proteins that are made in the ER is that it can become glycosylated, and that causes it to go to either the lysosome or the plasma membrane. One last thing that could happen to proteins that are made inside of the ER is that it could get folded in a particular manner, transported over to the Golgi apparatus, where it's modified even more, and then it's secreted from the cell. So this kind of goes along with what we talked about again earlier in the quarter. Remember we said that proteins that end up getting secreted from the cell, they're made in the ER and then the Golgi apparatus before it's secreted. And so here we're kind of adding a little bit more to that story. We're saying that there is a signal sequence that caused that protein to go to the ER, and then it's modified before it goes to the Golgi, and it's modified even more before it ends up getting secreted from the cell. So a third thing that could happen to proteins that are made inside of the endoplasmic reticulum is that nothing's added to it. No sequence is added to it. That protein then ends up going to the Golgi apparatus, where more modification is made before the protein ends up being secreted from the cell. So kind of the take-home point from all of this is that proteins have many different functions, which means that they're found in many different parts of the cell. They're even found outside of the cell, right? Some cells secrete proteins that end up doing things. In order for the proteins to get to their final destination, they either have specific signal sequences added to them, or they don't have signal sequences added to them. So again, the take-home point again is that the signal sequence, it kind of directs the cell where to deliver the protein.